Hello and welcome to this conference. We are glad to see you this morning. I just want to start with a little housekeeping for everybody so we can have a beautiful day together. So we're on Zoom. I think we're all very familiar with Zoom by this point. As much as we can, we'll keep everybody muted through the day so there's no distraction on the noise. But we do welcome interactions and questions. Please use the chat feature on Zoom. You should find that at the bottom of your screen. There will be many interesting speakers today. They will start with their presentation and then we will open the floor to questions. Please answer your questions in that chat box and we will address them at the end of their presentation. If there's anything through the day, I will be the tech support, so you can message me directly, or if you have a general questions, please feel free to add this to the chat as well. And I will pass it on to our host and MC for the day, Cynthia Brink.
Good morning, everyone. I'd like to welcome you all to our 9th annual research and conservation conference. We have quite a few interesting speakers today. I'd like to start with our land acknowledgement. The Long Point Biosphere region acknowledges that we meet on the traditional territories of the Adirondacks, the Haudenosaunee, and the Anishinaabe peoples, and show respect today to the communities of the Mississaugas of the Credit and the 6 nations of the Grand River Territory whose treaty lands include the Long Point Biosphere region. A significant portion of the Long Point Biosphere is located in Norfolk County on the shores of Lake Erie in Ontario, Canada. In 2018, the Long Point Walsingham Forest, which is the biosphere region, was selected by Environment and Climate Change Canada as priority place. It is one of 11 priority places identified in Canada and the only one so far identified in Ontario. It was selected because of its high biodiversity, large number of species at risk, and highly engaged local conservation community and significant environmental pressures. Today's presenters will be sharing some interesting research being done within the Long Point Biosphere region's watershed. We'll be starting with Michael McTavish. He is a MITEX Elevate postdoctoral research fellow working with the Smith Forest Health Lab at the University of Toronto and Ducks Unlimited Canada. His current research includes biological control of invasive plants and the ecology of non-native earthworms. Welcome, Michael.
Good morning. All right, are we getting started then? Perfect.
We are.
Okay. So I think I just need screen sharing there, which it's saying I don't have yet. Yeah, it's giving a message, can't share just because someone else is sharing right now, so maybe.
Yeah.
Okay.
Emily will share. There you go.
Great, thank you very much. Okay, let's put that up. How's that coming through? Perfect. All right, good morning, everyone. Thanks for joining us for the early first talk here. So we'll start things off right with some talking about Phrag. So I'm presenting work on behalf of myself and also colleagues Sandy and Rob from University of Toronto and Agriculture and Agri-Food Canada. And I'm going to be talking about the work we've been doing developing biological control for introduced Phragmites in Ontario. So if you work in the Long Point region, you're probably altogether too familiar with Phragmites. We're talking specifically about Phragmites australis subspecies australis, which is considered one of the most invasive plants in North America. It's thought to have been introduced from Eurasia in the late 19th century. The big problem here, of course, is it has a tendency to form these dense monospecific stands that can reduce biodiversity, alter various ecological processes, increase fire risk, and so on. It also can cause displacement of and possible hybridization with the native subspecies of Phragmites americanus as well. Control options do, of course, exist for Phragmites and can be effective in some cases, but they can also be limited in their effectiveness in others. They can be expensive and can pose various off-target risks as well. So for any problem of this complexity and scale, there's of course a critical need for additional control options to supplement what we're already doing. And that brings us to the idea of biological control, which is something that we like to think of as being another option in the toolbox. Biological control, or biocontrol, broadly is the use of live organisms to suppress an introduced pest. Why biocontrol? Well, weeds are often successful because they're introduced with fewer natural predators. So biocontrol is about reuniting that weed with its natural enemies. It ideally is something that can become self-sustaining as insects reproduce and track the host, which ideally makes it cost-effective at large spatial and temporal scales. Because great pains are taken to make sure that the biocontrol is very specific to the target plant, it's also very host-specific and therefore has low environmental impact on off-target species, unlike something like a broad-spectrum herbicide. Biocontrol of introduced Phragmites in North America is based on 2 European moths with stem-boring larvae shown here, so Arcanera neurica and Linisa geminipuncta. Both of these are natural pests of Phragmites reed beds in Europe. They contribute to Phragmites stem wilt and stem mortality as the larvae mine the tissues in the shoots as seen here. To give a general overview of the life cycle of both these species, which are very similar to each other, the larvae will hatch in the spring and feed in shoots for a period of about 4 to 6 weeks. So this is when you get the actual damage to the plant. They then pupate within the stems themselves, after which time they emerge briefly as adults and will mate, and ultimately lay several hundred eggs under the tight-fitting leaf sheaths of the plant, where they will be protected and overwinter until the following spring. Biocontrol programs typically take quite a while to implement, and so biocontrol for Phrag has actually been in development since 1998, when the initial proposals were put forward and explorations were conducted to find suitable agents. These were identified as the current agents by 2005. In 2007, then began the extensive process of host range testing to ensure their specificity on the target plant. This all culminated in a petition to release the agents in Canada and the US in 2018 that was then approved in Canada in 2019 by the CFIA. And that brings us to the present day, where we are conducting the first pilot program in Canada here in Ontario, including around the Long Point region. So I'm going to give a quick overview of various pieces of research that have happened since the summer of 2019 when I became involved with the project, and which is when this really kind of hit the ground at this pilot scale. The primary goal of all this work has been to implement this biocontrol at a pilot scale, actually on the ground with these 2 species. This is built around 3 main objectives, the first of which is to establish a baseline of background monitoring of Phragmites in the area. So this would include both the introduced and native subspecies to understand the system that we're interacting with better. The core of this is really about developing basic methodologies for rearing, release, and monitoring. So how do we produce the insects in large quantities? How do we put them out onto the landscape? And then how do we monitor how they are establishing and the impacts that they are hopefully having? Finally, by doing this, we're hoping to establish what we call preliminary nurse sites in Ontario. So these would be locations where the insects have an established population that we then can go back to as a source for other bugs in the future. In 2019, in the summer, this started off with this Phragmites survey of 63 sites across the southern part of the province. This included both the native and introduced subspecies. These were compared morphologically and with a genetic comparison to help practitioners and ourselves, of course, identify differences and how they interact. In 2020, in the spring, we conducted our very first releases. These were based on releases of eggs of both of the species. We were comparing 2 different release methods. One were a very basic one. We were just putting the eggs out on these cards that we would twist tie to stems to try and simulate the leaf sheaths on which they are naturally laid. And we also tried putting them into these screened cups, these inverted cups, to try and offer them some protection from the weather and from predators. Now, ultimately, because of how these eggs were overwintered, none of them actually hatched, which was unfortunate, but we were still able to learn quite a bit about potential threats to egg-based releases, namely predation. So we found when the eggs were out in the open, predation was very high. We lost about 90% of our eggs, and we found that the cups were a very effective means of providing some protection there. This past spring, 2021, was thankfully a very productive one, despite the challenges, of course, associated with working through the pandemic. One of the things we started off with was this overwintering hatch test. So once you have eggs from the previous years, one of the big questions is how do you maintain them over the winter? And so we compared eggs that were overwintered in different situations, so outdoors or indoor environments in North America. We also had some eggs that were sent very last minute in the spring from their overseas habitat. And we indeed saw that hatch could differ quite differently based on how you stored the egg. So we could get hatch that was as high as 95% with the right conditions, or as low as 37% in some cases as well. So this will help inform how we overwinter them going forward. So this is all about figuring out the best practices of how we do all these different steps. Another thing that we sometimes want to be able to do is to rear from eggs to pupae in the lab on diet. And one of our species, Arcanera neurica, will accept this diet. So we did a rearing experiment where we reared 372 eggs of the species on these pieces of artificial diet in the lab, and we were able to successfully produce about 65 pupae that we could then use for release in the field. This release actually happened in the Long Point region. So we put the pupae into a container, allow them to emerge as moths. In this case, we tried releasing in this large 6 foot by 6 foot cage that we have. The idea being to try and contain the moths so that they don't disperse too far before they are able to mate and oviposit. So we've since removed that cage, and we'll be returning next spring to try and see whether those eggs eventually produce larvae the following year. In contrast to that, because we expect that to work fairly well, but it is fairly labor intensive, we also tried some open pupil releases at 4 sites along a latitudinal gradient in southern Ontario. It's a very similar methodology, but without the cage. So we're trying to see how successful that will be and whether we can get away with skipping that particular step. We've also released several thousand larvae across several sites in Ontario. Our 2 main methods that we've converged on doing this with include the release of loose larvae, where essentially once they hatch, you just kind of dump them out onto the ground, which is very efficient and easy to do, but does produce fairly low larval survival. The sort of more involved method is to introduce them into these cut pieces of Phragmite stems. You can sort of shove a few larvae into the bottom and then put blocks of these stems out into the environment. This will give them a sheltered environment and food to get going before they then switch to other stems. This has worked very well so far, but it's also fairly laborious. So there's a bit of a trade-off there, and we're going to keep messing around with these and other alternatives. We had some extra pupae as well, so we did some cage tests at the University of Toronto, where we released pupae in cages at different densities to see if that had an effect. We found that there does seem to be a density-dependent response in terms of oviposition. So when you have too many moths, essentially you get lower oviposition, possibly because of pheromone confusion that's happening. They also tend to favor larger stems for ovipositing, which will help inform how we do cage releases like this in the future. So looking forward to next year, we're already getting ready to go with over 10,000 eggs that we have available for release next year. Essentially what we're going to be doing is more and more of what we've already been doing. So more experiments with storage, with rearing, with release. We're going to be trying out some new options, trying to refine the old options that we already have. Because 2021, this year, we were able to put out a bunch of larvae and pupae, a major focus for next year will be monitoring those release sites for signs of larval establishment. So we've seen feeding this year, but the big question will be whether they are able to overwinter and establish a population that will last for more than a year. So to wrap up, I just had a few general thoughts about biocontrol. Just as a reminder, this is about long-term solutions for large-scale problems. This isn't a quick tool, it's sort of a big picture tool that we hope to have exist alongside other options. What we're aiming for here is not complete eradication. This is about reducing the population with an additional pressure to combine with natural biotic resistance or other management tools. People are often concerned about the risks of biocontrol. That's why that host specificity testing takes so many years and is so thorough. It's also important to compare any risks that do exist against the risks of other management, which would include conventional tools, or the risks of doing nothing at all. And finally, biocontrol, though we think it holds much promise here, it's also not a panacea. This is, as we emphasize, another tool in the toolbox just to give us more options across all the different contexts in which we encounter this challenging invader. So biocontrol of introduced Phragmites has been in development for over 20 years. Again, this is a slow process, so it's really exciting to see it finally in this pilot stage in Ontario. We've now had insects here, we've reared them in the lab, we know how to produce them, how to put them out on the landscape, and we're already seeing promising early signs of larval feeding, including entrance and exit holes, stem wilt, stem mortality, the larvae themselves. So that's been really exciting to see this year. So really hoping that biocontrol is able to offer an effective, safe, and affordable new tool to help manage Phragmites going forwards. As always, these kinds of projects don't happen without the support of many different collaborators, so thank you to everyone involved in that. And if you have any questions, I'd be happy to address those now.
Thank you, Michael. We do have some questions already coming in the text box.
Mm-hmm. Did you want to read those out, or should I take a look at those? Sure.
I can read them for you. Yes. So Steve is asking, is the stem with the that ultimately kills the stem caused by a fungus? If so, do the control agent being used have the fungal spores?
Right, that's a very good question. So the primary mechanism of damage seems to actually just be the boring out of the stem itself. They're very thorough in their feeding, so you basically end with just a shell of the stem, and it will either die or spend enough resources branching out and trying to reinvest in growth that it doesn't really invest in reproduction for the next year. So that seems to be the main mechanism of action. But of course, because you do have insects boring holes into the stem, digging out the pith and so on, there is that potential for colonization by fungi and other pathogens. So that's not the main thing that we're looking for, but of course could happen, and it's something that we'll be monitoring for as well.
Great, thank you, Michael. Any other questions?
I know it's early, but happy to field any questions that anyone might have.
If you do not want to type and you want to take the mic, you are more than welcome to unmute yourself and ask your questions. I know sometimes typing can.
We're not scary here, I don't think.
There is another questions coming in, or comment. Thank you very much for a wonderful presentation. My question is, how will you find the same insect again in next spring?
Right. Yeah, that's a great question. So I don't know if it really comes across in these slides, but these insects they're pretty small, especially those first instars. They're really just a couple millimeters. So what we're not generally looking for is the insects themselves, but evidence of them being there. So the damage that they cause is kind of the best sign of it. So as the larvae develop, they will eat their way through one stem, leave, enter another stem, and go through 3, 4, 5 stems in this fashion. So there's a very characteristic pattern of damage and loss of the stems, but also those entrance and exit holes that they chew to switch between the holes. That's kind of the main thing that we're going to be looking for. And then you can, of course, open those stems up, look for the larvae, look for the pupal cases, and so on.
Awesome. John, the questions are coming in now. That's great. John's asking, what else do they eat?
What else do they eat? So they are very specific to Phragmites australis. That was sort of the focus of a lot of that host range testing, is to ensure that specificity, and that's what they lay on, and that's what they eat. So they're a monophagous species.
Steve is asking, how susceptible is the native subspecies to the moth larva?
Yeah, so that's a very important question, and something that we're keeping a close eye on. This was a major focus, again, of that host range testing because they are so familiar. The good news is that the native subspecies is really pretty safe from these. A lot of this has to do with the phenology of their development and some of the morphology as well. The big factor is that the moth eggs overwinter under the leaf sheaths. So sort of right where it connects to the stem, there's a sheltered area that they stay overwintered. And one of the big differences between these subspecies is that the native drops virtually all of its leaf sheaths in the late fall and into the winter. So even if any eggs are laid there, those leaf sheaths fall off, the eggs are exposed, and they die. So they won't overwinter until the next year. So we're watching that, but we're also not concerned. We have a lot of evidence to show that should be not a problem at all. Certainly not compared to the risks that introduced Phragmites already poses to native Phrag.
Amanda's asking if there's any threat of this species competing with native species.
Also a good question. Yes, so that is something that's part of the preliminary work that we've done on this. So before we actually did any of these releases, we've done some research looking at the insect communities that naturally occur on introduced and native Phragmites in different areas, the range of variation that's there. We, of course, can't know how the insect will impact them exactly in the field until they're actually out there, but we do at least know the baseline of that community, so we can monitor how it will change at some of these pilot sites. So that is something that we'll be tracking very closely in these early years.
Thank you. Victoria is saying she's not super familiar with the biocontrol literature, but as the host, specificity tests consider the effect not only ensuring they only eat the frag, but how native insects community is affected.
Oh, yeah. So I think that was the one I just answered, I believe, unless I'm losing it.
It was a similar question. It was asking you about competing with the native
yes.
species, but
so similar one, yes.
Yes.
There are others with some overlap in the feeding guild. So we know what's there, so we'll see how they respond once they arrive at these test sites.
Somebody else is asking if high water along the lake an issue. I think there was something like 100 years high last year.
Right, so lake water levels fluctuate on a semi-predictable basis along the Great Lakes Basin, and we know that is very important for how Phragmites succeeds overall. So we know, for example, that we're coming into a fairly dry period now, which is going to probably be bad in terms of Phragmites spread. So another thing that we'll be comparing is how these agents perform across gradients of water depth, for example. So when they're switching between stems, that's one of the big things that we'll be looking for. Right now, they can just, in a dry site, sort of walk over to another stem. One of the questions will be if they're standing water, how does that affect, for example, their dispersal or their ability to find a new stem? So we're going to have some sites that are along these gradients so we can compare efficacy between them.
Lots of great comments. Michael, thank you so much. Great talk. Looking forward to hearing how things progress. Interesting. Great. Thank you.
Okay. Thank you very much.
Thank you for being here.
Thank you very much, Michael. Our next guest speaker, Elizabeth Arango Ruda, graduated with a degree in forest engineering and a master's in hydrology at the National University of Columbia. Currently, she is a PhD student in Earth and Environmental Science working at the Hydro Meteorology and Climatology Group and the Center for Climate Change at McMaster University. She is conducting research in the biosphere and atmosphere interactions, particularly her study, she's studying the effect of climate variability and forest management activities on the water balance and age sequence of forest with an emphasis on transpiration. Welcome, Elizabeth.
Hello, thank you. I'm going to share my screen right now. Can you see it?
Yes, we can.
Okay,
bienvenido.
Gracias. Okay, so thank you. Good morning to all. I'm very happy to be here. So thank you for having me. So today I will be talking about the impact of climate variability on water exchanges in a deciduous temperate forest. This is the analysis as a result of my first year as a PhD student at McMaster University. So forests are globally significant for their capacity of providing benefits such as goods, services, livelihood support, ecosystem regulation, and terrestrial biodiversity. Particularly, temperate forests hold approximately 20% of the world plant biomass and 10% of the terrestrial carbon, providing important benefits for humanity, natural ecosystems. Due to this, it's important the protection and restoration of forests for adaptation, mitigation, and sustainable development. Also, restoring degraded forest lands presents the most significant natural climate solution. And it can be done by means of afforestation, reforestation, and natural regeneration. Additionally, temperate forests play a significant role in the global water balance. The vegetation influences climate through physical, chemical, and biological processes. Influenced directly the energy fluxes, the hydrological cycle, and the carbon cycle. Particularly, evapotranspiration is very important because it's the link between the energy and the water fluxes. Another important aspect of our research is the sustainable forest and management activities that refers to the management of the forest, keeping the balance between ecological, economic, and sociocultural pillars. Also, the variable rotation harvesting as a strategy to minimize the impact of logging operations, preserving the environmental values of the forest. So our research is conducted at TPV site, which is a temporary mixed deciduous forest of 90 years old. The site is managed by Long Point Region Conservation Authority, and it's part of the Turkey Point Observatory Station. So here you can see like, the location of the forest. The picture on the top shows the cover, the cover of forest of TPV. This is extracted by the Global Forest Watch. And there are also some pictures of what we have done in the field. Sorry, I can. Okay, this site is very interesting because it's a mixed forest, so the degree of biodiversity is higher than the other sites that we have been studying. The dominant species is Quercus alba, but there are also secondary and undersized species such as Acer saccharum, Acer rubrum, Fagus grandifolia. The density of the trees in this site is 504 trees per hectare, and trees have approximately a height of 25.7 meters. Our research is based on the covariance technique. So we are measuring half-hourly fluxes of momentum, latent heat, sensible heat, and CO2 using a closed path eddy covariance system that has a 3D sonic anemometer and infrared gas analyzer, as you can see in the pictures at the bottom. And we also have a data logger, that is the system that retrieves the information or the data that we are manipulating. I'm also showing some pictures of the site and the tower in winter, summer, and fall. For our research, we wanted to understand how climate variability is affecting evapotranspiration, volcanic conductance, that is the moving of water through the canopy, and the water use efficiency of the forests. So these are some of the results. So basically, we wanted to understand how photosynthetic active radiation, temperature, vapor pressure deficit, volumetric water contact, and precipitation are affecting those variables: evapotranspiration, canopy conductance, and water use efficiency. So here I'm showing the interannual variability of these climate factors. So the first figure is showing the photosynthetic active radiation. Second picture is showing the temperature of the air and also the temperature of the soil at 5 centimeters depth, and the temperature of the soil at 100 centimeters depth. Also, the interannual variability of the vapor pressure deficit, the volumetric water content, and the precipitation in the site. So here we have the cumulative curves of the precipitation, evapotranspiration, and precipitation minus evapotranspiration through the study period that was 2012 to 2020. So we also calculated the a drought index to identify what were the years where the there was like a moderate drought or the precipitation was lower. So the index tell us that the drought year was 2012 and 2016. And we can see that for those years, the amount of precipitation was also lower. But very interested is that even though the precipitation was low in those years, we see that there is no much variability in evapotranspiration. Even in 2016, evapotranspiration looked higher than the evapotranspiration, for example, in 2013. In the small boxes of each plot, you can see the annual cycle of the precipitation and the evapotranspiration. And we noticed that the precipitation was higher in 2017 and 2018, and was more evenly distributed during the months in 2020. This is the diurnal cycle of these environmental controls and the evapotranspiration and the canopy conductance. So as we saw in the last figure, we noticed that there is a clear difference in the environmental controls between in the years 2012, that is the year of drought, and 2019, that is considered a wet year. So photosynthetic active radiation, temperature, vapor pressure deficits are higher in 2012 than 2018. So that's probably what is impacting the increase in evapotranspiration during those years, because evapotranspiration is highly related to the temperature of the air. But volumetric water content and the canopy conductance are lower in 2012, and it has to do with the fact that there's no much water availability for in the soil or for the transport of water through the canopy. This is the relationship between the evapotranspiration and the canopy conductance and the vapor pressure deficit. We know that the vapor pressure deficit is highly affects or control the evapotranspiration and canopy conductance. And we can see the relationship. There is like the evapotranspiration increase as vapor pressure deficit increase. But we see that there are smaller values in 2012 because of the lower water availability. But the difference is not really huge. And for the canopy conductance, we see that there is less dispersion of the values of canopy conductance in the dry year. So it's less affected, it's constrained during the drought years compared to the values in 2019 or 2020, that are the years that are considered to have more water or have more precipitation events. And finally, we wanted to try to understand how the atmospheric demand and the water availability are affecting the canopy conductance, the evapotranspiration, and the water use efficiency. So that's why we try to separate the volumetric water content, like less than 0.07, and the data also for volumetric water content with it's higher than 0.007. So we see that our values they are less dispersed and are more consistent when we have lower soil moisture, and when they are also related to the vapor pressure deficit. When we have more water in the soil, there are more dispersion, values are have a higher range. Also, we see the clear relationship between each of the variables with a vapor pressure deficit. So we see that the canopy conductance decrease as BPD increase, evapotranspiration increase as BPD increase till certain point. After certain point of or value of BPD, there is no evapotranspiration, or the evapotranspiration is constant. And the water use efficiency also decrease as BPD increase. So basically, the vapor pressure deficit is one of the main controls of the movement of water to the atmosphere. So these are some of the conclusions. During the growing season, evapotranspiration responded positively to vapor pressure deficit, temperature, and photosynthetic active variation between regression lines of different types. Water use efficiency decreased with increase in atmospheric dryness, particularly in 2012 and 2016, that are the drought years. There was a slight inverse relationship between evapotranspiration and volumetric water content. Variation of evapotranspiration is conservative. It doesn't change significantly during the study period. The water balance, like precipitation minus evapotranspiration, was positive for all years, but small, particularly in 2012 and 2016. That are the years where there was less precipitation. And further research is required to understand what is happening to the stream flow in drought years when evapotranspiration is high due to high temperatures. So I want to thank to all the organizations that are giving us the support and the funding for following our research. So that's all. Thank you.
Thank you very much, Elizabeth. That was a lot of information in a very short time. We are opening the floor up to questions. Any questions for Elizabeth are very welcome.
I hope I was clear.
Yeah, it's a lot of information.
Can I ask a question?
Yes, of course.
I was wondering if you could tell us which result or which graph you found the most interesting or the most surprising.
Okay, so there is the topic about how atmospheric demand, water availability is affecting the forest. It's a lot of researchers are trying to understand what's happening there, because it's difficult to separate the effect of vapor pressure deficit and volumetric water content. So I will say that this is the figure that interests me the most, and is the one that I would like to continue to do further research, because it's difficult. Like as you can see in the figure, it's difficult to separate the effect. There is some change, some difference, but it's not clear. So I will say this is not the one that surprised me, but it's the one where I want to continue going deep and trying to understand better.
I have a follow-up, but I can wait and someone else has questions.
Go ahead, Jonas.
Okay. Yeah, I was wondering, so the Flex Tower you use, I mean, they're in a national and international network, right? There's a bunch of them in Canada and even more in the US. Will you be using data from other towers as well in your research, or just focusing on this one?
Right now, I'm just focusing in our sites. We have 4 sites. Sorry, 5, because we have 3 towers in the age sequence of White Pine Forest. This site that is the deciduous forest and an agricultural land. So far, I will be working in our sites, but if I see that there is data from the Phragmite team that is useful for my research or for comparisons of how water exchanges are depending on the forest biodiversity, forest structure, and composition, I will use the data.
Thank you.
You're welcome.
Thank you so much for your questions. We are a tad bit ahead of time today. Let's see if we have our next presenter ready to speak. Her name is Hadil Elsayed. She is a student, a PhD student in biology, working with Dr. Sheila Kolas, Native Pollinator Research Lab at York University. Her current research is a multi-collaborative project taking place within the Long Point region that focuses on the impact of climate and landscape changes on insect communities within the protected areas. She also is studying how long-term changes in insect communities impact aerial insectivores. Hadil's research includes modeling current and future changes in habitat suitability for at-risk insects species within the Long Point World Biosphere region. Welcome, Hadil.
Hi, everyone. I shall share my screen. Let's see. Okay. Perfect. Can you see the actual presentation, not the presenter side?
Yeah, we're good.
Perfect. Okay, I will start. Good morning, everyone. Thank you for being here. I will be presenting my research on the impacts of anthropogenic threats on insect biomass in the Long Point region. My email is down here in case you want to reach out to me later on for further questions. And I'll get started. Okay, so insects are very important, small but mighty. They are essential to life on this earth. They provide humans with ecosystem services, and these are functions we as humans obtain from ecosystems that are beneficial to us and are critical to our quality of life. Some examples of these services are pollination, where at least 1,500 agricultural crops rely solely on insect pollination. Without this pollination service that is provided to us by insects, food security would be threatened. Another example is pest control through the means of integrated pest management. And one more example is nutrient cycling, which is another service that we rely on, where herbivore insects are able to break down detritus and release nitrogen and phosphorus back into the soil for reuptake by plants. So insects also play an important role as bioindicators of ecosystem health because of their host plant specializations. Butterflies and moths are good indicators of the environmental quality. Another example would be dragonflies, caddisflies, stoneflies, and mayflies. They all depend on bodies of water, typically running water, for development during their nymph stage. During this stage, they tend to feed on other aquatic organisms, as well as mosquitoes. So besides being a good control for nuisances like mosquitoes, they are excellent bioindicators of wetland health. So even though insects are extremely important to us, they are facing worldwide declines. So a study by Hallman et al. Down here that took place in Germany showed that over a 27-year period, insect biomass decreased by more than 75% within protected areas. So another study also taking place in Germany over here by Siebold et al. Showed that within grasslands and forested areas, both insect biomass and diversity decreased over a 10-year period. So further studies have indicated that several taxa worldwide have declined as a result of increasing environmental stressors. Some of the largest declines of biodiversity have been seen in a 50-year monitoring of Lepidoptera species in Sweden, where 45% of these species were experiencing rapid declines. So insects are far less researched than mammals and birds. Interest in insects are far less compared to like the more charismatic megafauna. So over here, you can tell that arthropods are the most diverse phyla. They make up the largest percentage. And yet the IUCN Red List only lists about 0.5% of all described arthropod species. And the research that is being conducted on insects receives a lot less funding compared to those done on vertebrates. So if you look at this graph, you can tell that arthropods down here receive the least amount of funding per species compared to all the other phyla. So protected areas are important habitats. They come in various forms, such as national wildlife areas, national parks, conservation reserves, and natural reserves. They provide habitat to endangered species and are unfragmented natural ecosystems. They act as a refuge for many of the world's insect species. So protected areas are also important for tourism, biodiversity conservation, social and community objectives, as well as the aforementioned ecosystem services. So this here is a map of the various marine and terrestrial protected areas found worldwide. So terrestrial and protected area and marine protected areas make up about 15.7% and 7.9% respectively. With the lack of public interest and funding given to insect research, this is a rather small amount of land dedicated to protecting insect diversity when they're facing such large worldwide declines. So for this reason, I've chosen to focus my research on addressing how insect communities within protected areas has changed over a 30-year period and what environmental stressors underlie these changes. So my objectives for this research are to assess changes in the insect community composition, identify impacts of environmental stressors on insects, including weather, vegetation, and land use changes, and understand the role of interannual variation. So some historical information about my research. So my research stems from previous collections that occurred in the 90s. So in the 1990s, several Malaise traps were set out across the Long Point region by Peter Carson to collect flying insects. These samples have been in storage since then. In addition, vegetation data has been collected since 1991 by Michael Bradstreet and a few others. So my research is a two-parter. The first parter is an exact replication of the historical sampling that occurred in the 90s. So here I'll be sampling each of the sites using Malaise traps only for the same number of days that were sampled historically. This will happen throughout the 2021 to 2023 field seasons, depending on each site. The second part of my research is focused on interannual variation. So I've selected a subset of my sites that will be sampled for prolonged periods. These sites will have malaise traps set out all season from May to August, from 2021 to 2023. In addition, they'll have bi-weekly observation and sweep netting of Hymenoptera, which are the bees and the wasps, as well as Lepidoptera, which are the butterflies and moths. So with the ongoing pollinator declines, we wanted to focus on these major pollinating groups. In addition, butterflies and moths tend to lose their scales when stored in ethanol for the malaise traps, so they're harder to identify to species level. So this additional sampling will help us create a database for any future studies. So this is a map of my study sites. The red sites are all the sites that are only going to be sampled for the historical data, whereas the blue sites are the sites chosen for the prolonged sampling with the increased malaise trapping over the 3 years, bi-weekly sweep netting, and observations. So all 13 of my sites are located within protected areas. These 2 up here are located within the St. Williams Conservation Reserve, and the remaining 11 are located throughout the Long Point Peninsula, mostly within the National Wildlife Area. So for this year, I was able to sample 7 out of the 13 sites. 4 of them are the blue sites that are being sampled every year, and an additional 3 red sites that were sampled in 1992. So not all the sites were sampled every year in the 90s, so we'll be comparing samples from year to year. So methods for my sample analysis will include looking at both wet and dry biomass of the collected insect orders. These orders will be identified to the family level and whenever possible to the species level. So I just wrapped up my first field season, and I'm only presenting the data that I have ready. So all of this is very preliminary and will likely change in the future as I process more samples. So these graphs are looking at 2 of my 7 sampled sites. The first one is ROIS, which is the red oak iron savanna. And the second is RARO, the red ash red oak site. These are both located on the Long Point Peninsula. So if you look at the ROIS, you can tell that it is showing some decline from 1992 to 2021. Whereas if you look at RARO, it is showing albeit a bit unexpected, a steep increase in insect biomass. So if this is a bit unexpected, these 2 sites have comparable climate conditions. So my insight would be that this increase is being driven by vegetation or landscape level data. So this is still preliminary. I'm not sure. I won't be able to tell if that's the case until I start analyzing all the data. So if we take a closer look at each of the sites, so here for the ROIS site, most orders are a bit comparable between both years. But if you look here at the Lepidoptera group, it is showing a steep decrease in biomass between the 2 years. So this site showed a total of 13 orders collected between the 2 years. So look at the red ash red oak site, shows that Diptera is the order that's driving the increase in biomass we see at this site. This site shows a bit less order diversity compared to the first, with only 8 orders collected between the 2 years. So if we remove the Diptera group from the red ash red oak site, things tend to clear up a bit more. We start to see that Lepidoptera here in blue is showing a bit of a decline between both years, but we also see certain groups pop up that were not collected historically, were not found historically at this site. So here in orange, Hymenoptera wasn't found at the site in 1992, and neither was the Trichoptera group here in brown. So some interesting results from my data so far is that Trichoptera, or the caddisflies, weren't found at either site in 1992 but were found in samples from both of these sites in 2021. So this is a very interesting result, albeit still preliminary, and overall findings might change as I start analyzing samples from the remaining sites. These insects are very sensitive to climate change. So the observed increase of this group might be related to the vegetation and landscape level drivers. However, I won't be able to verify this until I start incorporating all the environmental stressors into my analyses. So my next steps for this research are to start identifying my samples to the species level and family level, as well as complete my 2022 and 2023 field season and assess all the environmental stressors, including climate change, vegetation, and land use changes. So I would like to thank everyone who has helped me with this project, from answering my questions about the historical sampling, the available data, to locating sites and site access, as well as permit applications. And I would like to thank my lab mates who were a sounding board for me during all of this. So thank you.
Thank you so much. Super interesting. Cannot wait to see more about your research. There's already a few questions, or at least one question popping in. We're welcoming more. Audrey is asking, will you be doing DNA analysis of the historic and new samples?
So I have a lot of historical samples and a lot more current samples that will be sampled. So DNA analysis is very expensive. So I'll be focusing on species level identification for now, but I am looking into doing environmental DNA analysis on the ethanol samples themselves, rather than turning all of the captured biomass into a mush and sending that out for analyses.
Thank you. Jonas, I think you have your hand up. If you want to unmute yourself, if you still have questions, you're more than welcome.
Okay. Yeah, I don't know. I like to talk. Great presentation. I was wondering if you are in contact with the person who did the 1992 sampling. One of my first thought was what if is the difference a difference in maybe in equipment or time you went out, or just maybe that person wasn't as interested in RNA or certain groups? Or
so the person who did all of the historical sampling is Peter Carson. I am in contact with them.
Cool.
The Malaise traps that I'm using now are a bit different from the ones they used back then, because the ones they used back then were specifically made for that project, and the traps no longer exist. Whereas the ones I have now are mass produced. And when it comes to timing, because of the nature of my study and that a lot of the sites are located on the National Wildlife Area, timing and access to sites differs day to day, but I try to keep the timing of when the traps get emptied consistent as possible.
I guess once you have more results from more sites and longer, that might sort of mellow out the difference, probably.
Yeah, so this is just looking at 2 sites. So hopefully when I start looking at the remaining sites, things will make a lot more sense.
Thank you.
All right, next question from Val. Given the small area of protected land around the world and in Canada, would it be useful to increase native gardens and small scales, neighborhoods, public lands, etc.?
Oh, definitely. So interest in insects, as I said, is very low. So a lot of the research or a lot of the weight is put on these protected areas to uphold the biomass and diversity of the species. So having more native plants, even if they're in your backyard, is a great way to preserve this biodiversity.
Awesome. Sam, who's a fellow researcher, has a question. She says, such an interesting presentation. Maybe I missed it, but what is the mass of the samples that you collected in the last field season? I found it was particularly low yield season fruit collection.
So I don't have the total mass of all my collections for this year, but for the two sites I did present, one of them was based on dry mass was 58 grams, and the other one was 80 grams. So a bit low for two sites that were sampled for almost a month.
We have another question. Thank you for really interesting presentation. Are you using the same methods and instruments which were used previously in the historic research?
Yes, so same exact methods, same instruments. The only difference is the malaise traps that I'm using might be a bit different. But I can't tell for sure because those malaise traps aren't around anymore to compare the net size or like the mesh size of the traps.
We have another question from Audrey. I may have missed it, but what environmental stressors are you including in the analysis?
So I have climate data, vegetation, and land use changes. So for climate, I'm looking at minimum and maximum temperatures, precipitation, wind, as well as number of frost days from the previous year for each of the sampled years. Vegetation level, so everything from shrubs, trees to plants. And then land use changes are both like landscape level drivers, including, especially for like the Long Point Peninsula, how high the water level was. And for the St. William Conservation Reserve, because there's a lot of farms around it, including those as well, and roads, all of that data into my analyses.
Any other questions? You can either put them in the chat, or if you want to raise your hands or unmute yourself, you're more than welcome to ask.
Yeah, I have a quick question here.
Go ahead, Carrie.
Thank you. I'm just thinking of the site, I can't remember the name of it, that had the increase in dry biomass. I know you haven't gone down to the species level yet, but what are the chances that it's an increase in the kind of quote unquote bad insects? So the insects that are typically found in areas that have a lot of human activity. Whereas if you looked at the species that are found in kind of good, healthy habitats, then you'd actually see a drop in that biomass.
I mean, that's likely, but the red ash, red oak site is located like right in the middle of the Long Point National Wildlife Area. There's very little human interaction there. So I'm not sure if that is a possibility or not. It might be a while before I get there.
Yeah, absolutely. Will you be looking at kind of the diversity of that biomass as well as some sort of an indicator?
Yes, so next step of my research is to take all these orders and start identifying them down to the species level whenever possible, depending on the keys I have available to me.
And does that include the 30-year-old samples too? Like have they
Yes.
been identified down to species level, or you're taking these old specimens and trying to do your best to?
I am taking these very old specimens, a lot of which have dried out by now, and identifying them down to species.
That's going to be enjoyable. Yeah, have fun with that.
It's going to be a long process.
Make sure you get yourself a couple of good lab assistants.
Yes, I am on the lookout for those.
All right, thank you very much.
Any last question? We're still running a little bit ahead of time. So if anybody else has anything, comments or questions. Oh, I see Sherry has something in the chat box here. So great to know that everyone can help by planting native plants in our yard, schools, whenever we can. Awesome. Great comment, Sherry. Awesome. Thank you so much for your presentation. We wish you the very best in your research.
Thank you very much. We are set to have our next speaker, Samm Reynolds. She is a graduate student at the University of Guelph in the School of Environmental Sciences and is a member of both the RAIN and the Young laboratories. Her research is focused on pollinator conservation on agricultural land in Norfolk County, with a focus on flower flies and native bee species. Sam is extremely passionate about educating others on the diversity of pollinators that we have here in Canada and why they're so important to our agricultural systems. Her work is being conducted in collaboration with the Canadian Wildlife Federation, Agriculture and Agri-Foods Canada, and she is extremely grateful to be receiving funding from MITEX, CWF, and OGS to support her work. Sam is looking forward to this presentation about her past field season in Norfolk County and what she hopes to glean from her data in the coming years. Welcome, Sam.
Hello, it's great to be here. I'm just going to figure out if I can share my screen. Okay. Let me know if you can see my presentation.
We can.
Yes, Sam, let's
Perfect. All right, so I will jump right in. Thank you for that introduction. So my name is Sam. I am a graduate student at the University of Guelph, being supervised by Dr. Andrew Young and Dr. Nigel Rain. This is a great presentation to follow the last presentation because there's some similarities in methodology and also in the goals of our work, as well as the volume of specimens that we have to get through. So today I'm going to talk about native pollinator conservation, specifically on agricultural land in Norfolk County, with a focus on flower flies and native bees. Some background, I'm sure all of you know how important pollinators are to our agricultural system, but I thought it would be good to give some background because I'm consistently astounded by the statistics. About 90% of flowering plants and 35% of food production relies on animal pollinators. So animal pollinators being bees, flower flies, beetles, birds, any sort of pollinating animal. And that equates to about 1 in 3 mouthfuls of food that you take. That number is debated a little bit as it's hard to get an exact, but for these purposes, that would be roughly 1/3 of the food that we have, specifically providing us with important vitamins like vitamins A, C, calcium, and folic acid, for examples. But not only are they critical for our health and food consumption, they also have a huge economic value. So the economic value of pollinators is somewhere between $700 billion Canadian a year to 1 trillion, which is a huge number, kind of unfathomable. But that was a paper came out in September reevaluating the valuation of pollinators, and they estimated it at roughly $1 trillion a year. So basically, all that to say, hugely important. Like a few people have mentioned, pollinators have been on the decline in recent years, and that's due to a number of different interacting factors like habitat loss and fragmentation, pesticide use, and climate change specifically. And my research looks at habitat loss and fragmentation. All right, so before I jump into my work specifically, I wanted to talk about the honeybee.
Sam, sorry, can you just move to a presentation view? A few people are commenting that we're seeing your notes and not the whole presentation.
Are? Okay.
Thank you.
Absolutely, yeah. Sometimes with this screen share, I don't know which one you can see. Can you see just my presentation now?
Perfect, thank you.
Okay, awesome. Yes, so maybe I'll just jump back. So honeybees specifically, when I say pollinator, a lot of people think about honeybees, but they're actually not native. They have a bit of a research and media monopoly. And so I am not focusing on them in my research. Honeybees are a managed species, so they're brought into Canada and they're rented out primarily as livestock. No shade to honeybees, they're incredibly important to our agricultural systems, but they're not the focus of my research for quite a few reasons. The main one being that right now our agricultural demands actually outstrip the rate of growth in honeybee stocks. So we are losing honeybees due to a bunch of different factors. And so everything that challenges that honeybee is then a direct risk to our food sustainability and our food security. Not to mention that they can be expensive for farmers. A lot of farmers have to supplement their pollination services by renting out hives. So you need about 1 to 3 hives per acre of land. And that can cost anywhere between $75 to $200 per hive. So with a big farm, that adds up pretty quickly. And there's also been some recent studies that have shown that honeybees can compete for resources with our native bees, which obviously is not ideal. So native bees, our native bees provide our pollination services completely free of charge, which is a huge bonus there. There's also over 800 species of native bee in Canada. And a lot of people aren't able to recognize them or don't know how diverse our native bees are. So that's something I'm super passionate about. They've also been shown to improve seed quality, commercial value, and shelf life for tons of different crops here in Canada, like squash, blueberries, apples. Recently it was Halloween, so all of those pumpkins we can thank the hoary squash bee for. Also, our native species have co-evolved with our native crop species. And so a lot of them are actually specialized to pollinate those crops specifically, which makes them a lot more effective than the managed honeybee. They can be up to 300 times more effective than honeybees. Specifically, I'm thinking of the genera Osmia, and they're incredible pollinators for specific families of crops. And so for all those reasons, I think it's really important that we focus some research on these native species. The other group that I'm looking at for my work are flower flies. Flower flies are substantially understudied and often mistaken for bees, but they are flies. They pollinate more than 100 cultivated plants, and they can also provide the pollination services that we need for over a third of our canola fields, which is pretty amazing. The other really cool thing about flower flies that I'm really excited about is that they can be used for integrated pest management systems. So they can be used for pest control because they have sort of a dual life history. So when the flower flies are in their adult stage, they can provide the ecosystem service of pollination. But when they're in their larval stage, they can actually be predatory, and a lot of them eat aphids. So that's obviously a pest that needs controlling. And so that's a really cool part of their life history. Okay, so like I mentioned at the beginning, my research focuses on habitat loss specifically. That's been happening a lot in the past few decades. And what we need to understand is that native insects nesting and foraging requirements aren't really well understood. We don't know particularly what each species needs. And so that makes it really difficult to create policies and to advocate for conservation efforts because we're not really sure what we should be advocating for. What we do know is that there has been declines in pollinators due to things like monoculture and intensive farming. We've seen in recent years a lot of hedgerows being ripped out and forests being taken down to make more room for agricultural land. And we don't know how that's going to impact our pollinators because we don't know what habitats they're using most frequently and what levels and what connectivity we need for them. There has been some studies conducted in the US and Europe that has showed enhancing natural habitats does support pollinator populations, both their abundance and diversity, which is great. But a lot of those studies basically say sort of pollinators or bees, and they leave out flower flies and they don't go deeper than often family level. So that makes it difficult to make very specific policy advocations because each species is going to have a different need. And so we need more large-scale habitat studies that actually go down to species level so that we can understand the patterns, not just for bees as a whole, but for each specific species, because we're finding that their needs are so diverse, especially with bumblebees. Bumblebees are quite well studied compared to a lot of the other native bees. And we're seeing that some are increasing over the last few years, while others are on the decline quite drastically, and it's not really well understood why that is. So these studies are very important, and I think once we understand that, we'd have opportunities to design agricultural landscapes in a way that would promote pollinator diversity and abundance. All right, so the habitats of interest. For the purposes of my work, I'm looking at 3 different habitat types that I thought would be beneficial for pollinators. So I'm looking at forest patches, hedgerows, and prairie grass to see how these 3 habitats affect the abundance and diversity of our native pollinators. My hypothesis specifically is that insect pollinator species on agricultural land would benefit from the proximity and connectivity of those 3 habitats, with forest patch being the most vital habitat for abundance and diversity. And my objectives are sort of threefold. So I want to assess the difference in pollinator diversity and abundance, both among the 3 habitat types. So on a large scale pattern, which habitat seems the most beneficial for diversity and abundance, but also among the patch sizes. So what size forest, what size prairie grass patch, and what size hedgerow seems the most effective, or does it have an effect? And then the last objective would be to assess the difference in diversity and abundance among patches with differing levels of connectivity to other pollinator habitats. So I want to look at the landscape composition and see how the connectivity of the habitats actually comes into play as well. So why Norfolk County? I'm sure we're all here for similar reasons. Norfolk County is a pretty amazing place to study because it has a lot of biodiversity. When I was first looking into where we should conduct this research, I found that that Norfolk County is called Ontario's Garden, which is very accurate. There's a ton of different diversity in agricultural crops like ginseng and cherries and asparagus and squash and lavender and all these other things that make it a really interesting place to study. The other thing that was really appealing to me was that there was a substantial forest cover compared to a lot of other agricultural areas. So about 18 to 29% from my last look for forest cover. Whereas I also had some traps set up in Harrow, and that had about 5 to 7% forest cover. So quite drastically different in 2 different agricultural areas. And that was really interesting to me. Another thing that I found really cool is one of my mentors, Dr. Jeff Skevington, who is a birder, and many of you may know. He also studies syrphids, also known as flower flies, which are my fly species of interest or fly family of interest. But he had noted that it could be a potential stopover spot for migration of syrphids. So had seen some mass migration of syrphids in and among Norfolk County, similar to how it is a stopover spot for birds. So that was also a really interesting component for me because I wanted to see how the abundance compared to our other study site, which is in Harrow. Another really appealing part of Norfolk was programs like ALICE, which are already in place and provided a lot of different diverse habitat types on many farms. And it made it a lot easier to find farms that fit the specific criteria of my study. So my methodology, we've all heard about Malaise traps already. They're basically tent-like structures that intercept insects' flights and direct them up into the head of the trap, which is filled with ethanol to collect our samples. So I just completed the second field season for this project this past summer, and we did an 8-week collection period. So I had 60 Malaise traps total. 48 of those were set up in and around Norfolk, and 12 of those were set up in Harrow. The difference there being because it was really difficult to find sites in Harrow that actually fit our criteria. So I was looking for farms that had all 3 habitat types of interest on the property, and also that each habitat was a specific distance away so that there wouldn't be a ton of overlap between the insect specimens' flights from each habitat type. So we did collect every 3 days. We went and emptied the Malaise traps. And so we collected them, every 3 days for 8 weeks. And then I also went out and did weekly transects because Malaise traps are sort of a passive collection method. They can have biases. So I wanted to make sure that there wasn't any specimens that we were seeing when I went out to observe and to do targeted transects that weren't getting caught in the Malaise traps, and just to sort of round out the scope and make sure that I was seeing all of the diversity in the area. And then right now, I am currently going through the process of identifying all the pollinators that we collected from the Malaise trap to species level, which is no small feat. In the bottom corner here, you can also see sort of a comparison between the Norfolk County and the Harrow site. around a 1,000-meter radius around our sampling site. So you can see that there's many more row crops in Harrow, much less forest, and overall just less diversity in the landscape. And so that'll be really interesting to compare the biodiversity of insects in both of those spots. All right. So the next stages of the project are to use taxonomic keys to visually identify all the specimens. So as I said, this is my second field season. For the first field season, I have already identified about 5,000 flower flies to species level and about 18,000 native bees to genus level. And I'm working on getting them to species right now. For this past field season, I have sorted all of the malaise traps with some help from some amazing summer students. So all the pollinators have been pulled from the malaise traps, and I'm now in the process of drying and pinning and labeling all of those specimens so that I can start IDing them in the new year, which I'm really looking forward to. I'm also doing some DNA work, basically trying to create a DNA library for the flower flies that are not morphologically identifiable. So some of the female flower flies are indistinguishable between species, whereas the males are distinguishable. So using that, using those males, we can create a DNA library that then can aid us in identifying the females, which has been really interesting for me. And I'm really enjoying that aspect of the project. And then the last thing I have to do is obviously put all of it into a database and then start my statistical analyses. So it is definitely a big project and it's going to take some time, but I'm really excited about the results that will come. So the impact of the research, I think what's really cool about this project is that it's going to create a novel data set that has an inclusive focus on flower flies as well as every species of native bees that can be ID'd, because some of them, they're pretty rough to work with in terms of the keys. They sort of need to be reviewed. But yeah, I'm really excited about that. I'm also, a lot of current research is just kind of like blending up all of their specimens into sort of like a smoothie of DNA and then running it through to see what specimens are present. I really wanted to do this project differently to retain all of the specimens so that I could have an accurate count of abundance, but also so that all of the specimens would be pinned and could be referred back to and also used for future studies. So I'm doing this sort of the long way, but I think it'll be worth it in the end to have this data set. My goal with the project would be to inform government policy and NGOs on how we can boost native pollinator populations on agricultural land by designing the landscape in a way that supports them, and also by co-creating knowledge with the local farmers. I've had a really great time working with them and collaborating with them, and so I'm excited to keep doing that in the future. I also ultimately have the goal of conserving native pollinators, increasing crop production, which would of course come with an inherent economic benefit. And then some other benefits that I've been reading about that can come from designing our landscapes to support pollinators can also create more sustainable agricultural systems in general by mitigating soil erosion and improving water quality and nutrient cycling. So these are all obviously great things that can come out of having properly designed landscapes once we understand what the habitat needs are of our species that are using them. So I'm doing this work in collaboration with Canadian Wildlife Federation, as well as Agriculture and Agri-Foods Canada, and I've been really grateful for their support and mentorship on this. There's also so many people to thank for their support on this work. So I just wanted to take this time now to thank them all and take any questions that you may have.
Thank you so much, Sam. Wonderful presentation. Really appreciate all the data and the hard work that's going into this research. We have the floor open now for questions. You're welcome to unmute and ask questions or just put them in the chat. We have one comment here from Betty Cheney. This has been the most interesting, Sam. Thank you for your presentation. It has been great working with you.
Thank you, Betty. It's been great working with you. I was excited to see your name on there.
Michael, you have your hand raised. Go ahead.
Yeah, thanks. Thanks for the presentation, Sam. Sounds like you're going to be have your work cut out with you with all those samples too. So
yes.
that's great. I'm just wondering, I don't know, maybe you're early in the IDs here, but I'm curious with the land use change and invasion and all these other stressors, have you seen or do you have any predictions about potential community changes in terms of very generalist pollinators versus very specialized ones? Yeah, any thoughts on that?
Yeah, absolutely. There's been a lot of research recently that has shown how generalists are affected compared to specialists on land use changes, specifically sort of looking at the honeybee as generalists compared to some of the more niche native species. And it does look like generalists are being more supported, which sort of makes sense. Obviously, if you're providing a bunch of row crops that flower at one specific time, the generalists can go in and use those resources, whereas a lot of the native species who have specific needs aren't having those needs met. We've also seen what has been most interesting to me so far. I didn't put this in the presentation just because it's very preliminary and I want to make sure everything's statistically relevant before I start talking about it, but I've been looking at the samples that we collected from Harrow, which, like I mentioned, was a very low diversity in terms of habitat types compared to Norfolk. And it's been really interesting because there are like full bags of, this is basically what a Malaise trap sample looks like. But there's like this whole bag is filled with flower flies of one family. So high abundance, low diversity. Whereas in Norfolk, there's high diversity and low abundance is what I'm noticing as like a general trend. And so I'm very interested to like dig into that and figure out why that is, because I think that's going to be really interesting just to see the stark difference between those two locations and what could be causing that. But I am noticing some patterns already, which is exciting. I'll be interested to present on that once it's ready.
Interesting, thanks.
Yeah, no worries.
Okay, and I've got another participant here, Catherine. She says, thanks, Sam. Great information. Enjoyed your visits to our property. I was not aware that flower flies migrated. Can you share a sentence or two about where they go and when they move north and south?
Yes, absolutely. It is actually similar to birds. So not all flower flies migrate, but some do. Right now, I'm actually working with a professor in Calgary who's doing some research on the migration process. So there's one species called Allegrapta obliqua that's pretty common and they don't migrate. And then there's another called Eupiodes americanus that does. And so right now we're actually trying to see if there's anything in the DNA that would be able to help us to see which ones migrate and which ones don't migrate, which is really interesting to me because we still don't have a lot of research on where they're going and how often they migrate and which species actually do migrate. But there is some data on that. And usually it'll be around September. I think for the species that I'm thinking of, they migrate around mid to late September in southern Ontario and arrive back early, I want to say like May. But there's definitely still more research that needs to go into that. And I can, since I have your information, Catherine, I can send you some more information on that once I sit down and write something up for you, if that would be interesting to you.
Thank you. I see Jonas has his hand up. Go ahead, Jonas.
Yeah, another great presentation. Hi.
Thank you.
Figured I'd do the Michael thing and show my face. I had two questions. The first being like at the end of this, if you can dream a little bit, and when you're an expert, do you think it would be a good idea to do an updated key on flowering flies?
So the flower flies key is actually, it was just recently updated. I want to say maybe 2 years ago by Dr. Jeff Skevington and my supervisor, Dr. Andrew Young. And so that book is actually incredibly comprehensive and it's what I learned to do all of my IDs with. I have it here for a prop.
Nice.
But yeah, so this is the book and it's really amazing because flower flies are sort of one family. It's a lot easier to do those IDs than it is to ID native bees because obviously native bees, there's all the different families and then the genera and then the subspecies, whereas this is just sort of a book of genera and then each species. And the photos are really amazing in it and helps with IDs. So I think that this probably is taken care of. But I would love to do some
Right.
reviews on native bee species, because there are some there's some genuses that really need some help for the native bees. So I that would be something that I would be willing to look into in the future for sure.
I guess the other question was to do with the last speaker as well. You guys are doing similar trapping and similar, I don't know if they're, and I know Sheila Cola's students have done work before also in this area. Do you think there are synergies in you guys sharing data with each other?
Absolutely. I would be really interested to see if our findings align and if we're seeing similar trends. So that is definitely something that I'm hoping to learn as we both get our data processed, which will probably take some years, but eventually down the line. Yeah, absolutely.
Thank you.
Wonderful. Thank you so much for the questions. We have a couple comments here and a couple more questions. One from Sherry Shrub. She says, thank you so much for this presentation. So interesting. Then we have a question from Mandy Karsh. Is there a wish list of plants to plant along roadsides to help support pollinator species?
Yes, there definitely is a lot of resources online about that. And I would also like to contribute to that. I've read a couple papers recently that looked at bumblebees specifically and showed how important late flowering plants are, specifically in September. So a lot of clovers and things like that had been really helpful for bumblebees. I think the big thing is to make sure that there are flowering plants for the whole season. So making sure that there isn't a lack of resources near the end or near the beginning of the season. So that's a really big one. I also always recommend native plants, of course, because like I said, our native species co-evolved with our native plant species. So they actually usually prefer those species, whereas honeybees actually prefer a lot of the invasive or more exotic ornamental plants. And that can help to propagate their survival over our native species. So there is a lot of really great lists online. I think Canadian Wildlife Federation has a list, but basically, you know, any sort of like butterfly milkweed, milkweeds, any native species, I would recommend planting, and then making sure that there's enough late blooming species as well for those bumblebees.
Okay, thanks for the question, Mandy. Another question from Audrey. Why did you think that forests would be more influential than prairie habitat?
Great question. I personally thought that because of the nesting needs of a lot of bees. So a lot of native bees are solitary bees and they are ground nesting or cavity nesting bees. And so they need sort of leaf litter and open areas that are undisturbed. And so I thought that the forest would provide the best nesting habitats for those species. And we've also seen that with flower flies as well. Of course, the prairie grass is incredibly important for providing foraging. So I think they go hand in hand, but I was thinking more of nesting habitats when I said that. And if there's a lot of flowering plants in the forest, then that's all the better as well. And I did notice that most of our forest sites over the last summer had higher abundance and diversity of flower flies and native bees. So far, that's supporting my thought, but again, I'll need to wait for the results to be more, established before I make any claims about that.
Okay, wonderful. Thanks for the question, Audrey. We have one more person here who has basically made a statement with regards to needing more public education. Her name is Patrina. She's saying public education on the diversity of pollinators is so needed. I'd love to see some large signs in parks and or on trailheads based on this topic.
Yeah, I completely agree. That's sort of my my passion and where I hope to go into after I finish my degree is more on the education and policy advocacy side, because I think that the first step to conservation is to know what we need to conserve. And so a lot of people, when I ask them about what bees they know, it's sort of honeybee and bumblebee, and that's it. And it's, there's 800 different species in Canada. So that's something I'm really passionate about.
Yeah, that's very true. Most of us have honey on our breakfast table, and most people relate to just that. I have Bailey Bingham that has actually posted to everybody in the chat
awesome.
a beautiful link for check out the Grow Me Instead guide on the
Yes,
Ontario
that's great.
Invasive Plants website. Yeah. So if there are no more questions, a lot of great questions. Obviously, people are quite interested in insects. We will go to before we go to break, seeing as we're ahead of schedule, we're going to go to Mandy Karsh from Road Ecology Group, the Ontario Road Ecology Group. She has a public service announcement about some of the work that she's done. Thank you so much, Samm Reynolds. It's been a pleasure listening to you.
Thank you.
Thank you, and good morning, everyone. And yes, I appreciate the pollinator comments, because the road working group for the priority place, the Long Point Walsingham priority place, we are focused on protecting biodiversity from the threats of roads, and certainly all taxa, pollinators, vertebrates, they're all included. So right now, what we want to do first and foremost is the roads working group. We want to thank the Norfolk community. It's full of amazing residents who have been saving wildlife from the roads, collecting data, and have been doing this for decades. So first and foremost, thank you. And we just want to speak to this group because, you know, whatever brought you to this conference, we can all help protect biodiversity from the threats of roads. And we're here as a resource if you want to learn how to move wildlife across the road for your safety and the animal safety. And we encourage you to report your sightings. Carrie Gunson of EcoCare International works tirelessly to manage data and the Wildlife on Roads in Ontario project within the iNaturalist platform. And she's an amazing resource and very open and willing to help teach people how to collect data properly and safely, and how to then upload your data to the iNaturalist platform through the Wildlife on Roads in Ontario project. And we are here as a resource. Please know that whatever data you contribute goes right to Carrie, and almost instantaneously, she works very hard to translate those data points into maps that then inform conservation and mitigation on the landscape. We work very closely with Norfolk County, and they are very supportive in learning where these hotspots are so that we can focus our mitigation efforts and dollars to protect wildlife in those areas. But of course, with an extensive road network and extensive habitat, it really is a public endeavor to help watch for wildlife and move wildlife, and of course, report your sightings. So again, Carrie is a wonderful person and an expert in road ecology. Her email is there. You can call or text to learn about iNaturalist and the Wildlife on Roads platform, how to upload data. The conference has my contact information as well, as the Road Ecology Group. So welcome. We encourage anybody to participate in helping wildlife cross the road. And again, we just want to thank the Norfolk community who just works tirelessly, again, for decades, protecting wildlife on the roads. And we're just here to help support that and sort of make it, one area that we can report sightings and then inform mitigation with the county. So thank you, everyone.
Thank you so much, Mandy. So we are done our morning portion of our presentations for this conference. I appreciate your time and wonderful presentations.
We will be back at starting promptly at 1:10 this afternoon. I invite you to join us a few minutes beforehand to make sure you don't miss anything. Please remember that there's a different link for the afternoon session. You will also get an email reminder. I scheduled an email to go to you around lunchtime to help you get back on. So a different link. So I will stop recording this morning session, but I will stay on a few more moments. If anybody wants to chat to one another, or you're more than welcome to do that. And then we'll see you in a different link at 1:10 p.m.